Okay, it's 9 o'clock, so welcome to BME 3721. I did see a lot of familiar faces, from my data evaluation principle. Anyway, so this is a Friday seminar, and we have a special guest from University of Alabama, Birmingham, and Dr. Mary Catherine Sewell-Loftin. And so I'm going to read the introduction a little bit. So Dr. Sewell-Loftin is an assistant professor in the Department of Biomedical Engineering. She's also a faculty member within the O'Neill Comprehensive Cancer Research Center at the University of Alabama, Birmingham. So she completed her bachelor and a master degree in chemical engineering at the University of Alabama. And after that, she went to Vanderbilt. She did her PhD under the supervision of Professor David Merriman, who happens to be the PhD advisor of Dr. Hutchinson as well. So her PhD study focuses on the development, mechanics, and the disease of aortic valves, and as well as tissue engineering. And I'm going to quote Dr. Hutchinson. Okay, upon completing her PhD, she joined the dark side and transitioned to cancer-related research during her postdoc fellowship at Washington University, St. Louis. So welcome to the dark side, also not particular camp. So prior to joining the faculty at UAB, Dr. Sewell-Loftin also received the prestigious K99R00 award from NIH. So her current research uses microfluidic platforms to study cancer cell migration, with implications to understanding and treat metastasis. So she's going to give a seminar about her research work today, and please help me welcome Dr. Sewell-Loftin.
Thank you so much for the invitation. Dr. Hutchinson actually did invite me here. He was really excited, and then he was like, oh yeah, by the way, I'm going to be in Austria. So between that and a little comment about the dark side, if you want to know embarrassing Dr. Hutchinson graduate school stories, I'll be around all day. So I'm Dr. M.K. Sewell-Loftin. I actually did grow up in Birmingham, Alabama, so you may hear some southern accent every once in a while. I'll try really hard to keep that out. But it is always nice to visit other places in the South that appreciate that kind of thing. So today I'm going to present some of our recently published and ongoing work, particularly focused on the tumor microenvironment and mechanosignaling in VEGFR2. And if you're not super deep into molecular biology, tumor development, I promise I'm going to explain what that actually means as I go through this. So one of the only positive things I can say about being a cancer researcher is I don't have to spend a ton of time telling you that cancer is bad. Cancer is the second leading cause of death worldwide. The American Cancer Society actually estimates that your incidence of getting cancer is somewhere between 1 in 2 and 1 in 3. Which means basically 1 in 2 or 1 in 3 people in this room is going to experience some type of cancer within their lifespan. Doesn't necessarily mean it's going to be malignant, a deadly type, but some type of cancer. And over the last 40 to 50 years, we've gotten a lot better at our early detection rates. So we can find tumors earlier and earlier in patients. We've improved our screening. We've done a lot of education in patient populations to make sure that people know what to look for in themselves so that they can also catch it early. But as you can see from this graph, it's a few years old now. Overall mortality rates are still pretty flat. We haven't really improved the mortality at all. And 90% of all cancer-related deaths actually happen because of a metastatic spread. So when you're initially diagnosed with cancer, it's usually at a primary site. Lung cancer means the primary tumor is in the lungs. A metastasis then is when it moves to a secondary location. So that can be brain, bone marrow, liver. Those are kind of the common ones. And the problem with metastatic disease is that because these cells have escaped that primary location, they tend to become more aggressive. They want to proliferate more, they want to remodel their environment more, and that also corresponds to a decrease in these cells' response to chemotherapies. So often the drug that will work for a cancer patient in their primary site when they're first diagnosed will no longer work when that disease occurs in that same patient later. And as an engineer, I was like, okay, well, there's got to be something going on in the microenvironment around the tumor cells that explains this very dramatic change in cell behavior, so can we investigate that? And even though Josh calls it the dark side, I really started off in cardiovascular in the context of cancer. So angiogenesis is the process by which new blood vessels will grow from existing blood vessels. And this happens in cancer because as a tumor gets larger and larger, it eventually reaches a size where diffusion of oxygen and glucose can no longer sufficiently keep the cells in the center of the tumor alive. So when the tumor gets to be big enough, it will turn on a switch, and I'm a pointer, so I'm going to walk over here and gesture things. It will turn on a switch where it creates a communication pathway to local blood vessels to create a sprout. That sprout will then grow into new vasculature that moves into the tumor itself to provide nutrients, which means the tumor can get bigger. But it also is a highway then into the cardiovascular system so that these tumor cells are able to metastasize. This was first described in the early 1980s, and it was very exciting because we discovered that vascular endothelial growth factor, or VEGF, was a huge driver of this process. And all of the cancer researchers said, great, let's just give inhibitors to VEGF and its receptor, VEGFR2, and that should shut down all cancer growth. It doesn't. In fact, most cancers that can be treated with anti-VEGFR drugs receive those drugs plus combinations of chemotherapy, and they still only see somewhere between about 2 and 4 months of survival improvement. So I had done work in Dr. Merriman's lab at Vanderbilt on the idea of epithelial to mesenchymal transition as a mechanical feature, and this actually ties in really nicely with angiogenesis because when that sprouting capillary forms, there's the same kind of phenotypic shift. So the first step of angiogenesis is you have this nice quiescent normal blood vessel with endothelial cells just minding their own business. Whenever they are stimulated to grow into a new vessel, you get this tip cell, which is shown in green, that's highly migratory. It wants to move, it wants to remodel the matrix, and it's gonna pull those other endothelial cells along with it to create that new sprout. And the same kind of cell behavior shift that I had studied as a PhD student, EMT, epithelial to mesenchymal transition, is the same kind of shift that happens to create that tip cell. And so we knew that this could happen with interstitial flow. That's something that's been shown in developmental biology, so shear stress will make these blood vessels grow. We knew it happens with soluble factors like VEGF and HIF, which is hypoxia-inducible factor. And my question was, can other mechanical forces really drive this behavior? Can strain physically cause the formation of tip cells? And what in the tumor microenvironment can actually make those kinds of mechanical strains change these cell behaviors and change angiogenesis? So this is when, in Dr. George's lab at WashU, I encountered cancer-associated fibroblasts for the first time, or CAFs. If you're familiar with normal fibroblasts, they're your resident maintenance cells. So they don't really do a whole lot when things are normal and you're healthy. And they just kind of hang out in your extracellular matrix. Maybe do a little bit of remodeling, but nothing too exciting or crazy. When it becomes an activated fibroblast, or a myofibroblast-like cell, now it becomes more polarized. Its expression of alpha smooth muscle actin is increased dramatically. That's these red stripes going this way. It secretes a lot of collagen, and it will remodel the matrix really, really significantly compared to normal matched fibroblasts. There had also been some other work out of the Cindy Reinhardt-King lab that took patient-matched normal fibroblasts and breast cancer fibroblasts and put them on 2D gels and looked at traction forces. And what they found was in all of these cases, the CAFs had higher traction forces than the normal fibroblasts. But the range was pretty dynamic. So you can see kind of in part B, What's a normal force for one patient is higher than the normal force of another patient. But there's still a significant difference between these normal fibroblasts and the CAFs in every case. Too many buttons. Here we go. The other lab that I worked in, the Longmore lab, was looking at CAFs as well, and this shows traction force mapping for mouse breast cancer CAFs that have had DDR2 knocked out. DDR2, I will not be talking about more, I'll just tell you that it's a collagen receptor, so it's how the CAFs actually can bind to the collagen in the tumor microenvironment. And when you get rid of that collagen binding, you get rid of the ability of the CAFs to deform a matrix. So I really wanted to take all of these things together and study how do strains or forces generated in a 3D environment affect angiogenesis, and how does that then affect tumor progression. So I wanted to come up with a model that would actually let me do that. And from a cancer biology standpoint, our models exist along this continuum. 2D is the simplest. It's a tissue culture plastic. It grows cells in a monolayer, and it's super easy to kill cancer cells in that situation. Give it enough drug, they all die, 100%. The next step up is kind of the idea of co-cultures in 3D. So we create structures that look more like what might be present inside a body. The gold standard of preclinical models right now is mice. And if I told you that we've cured a bunch of cancers in mice, you would never know it because those cancers still exist in humans. This is because mice are really terrible people. It's just a known fact of the universe. And then obviously, we eventually want to take things into clinical practice. We want to be able to treat human patients, human disease, and help them. And this continuum increases in complexity as we go from left to right, but it also increases in time and money, and ethical considerations too, right? You don't want to do 1,000 mouse experiments if you haven't screened your drug in some kind of in vitro model. The problem with this is that it's an inverse relationship. So our ability to control what's going on in the system is inversely proportional to the complexity and the physiological relevance of that system. So all models are models, and some models are useful, and we have settled in this 3D co-culture system, and you're going back and forth between that system and some in vivo models in my lab. To further explore the idea of strains in the matrix, we developed a bead tracing protocol. I like to call this poor man's traction force microscopy because it doesn't take a high resolution confocal microscope to do it. But essentially, we're measuring matrix deformations that are caused by cells embedded in a 3D gel. So these are vector maps that trace the movement of 1 micron fluorescent beads over 20 to 30 minutes. NBF is a normal breast fibroblast, CAF is the patient-matched CAF fibroblast, a blue arrow is a small movement, and a red arrow is a really big movement. And we know how many fibroblasts, actually, so this one was for an hour, but essentially what that means is instead of looking at like the rapid, dynamic, immediate shifting of cells or long-term matrix remodeling, this is really a snapshot of how much force a cell can generate and how much it might affect its neighbor in some signaling pathways related to proliferation and growth. We can take those vector maps and plot them in a variety of ways. I was really into histograms at one point, so I made a lot of histograms. But all that lets you do is really see that the CAFs have a higher average magnitude of deformation compared to the NBFs, and they're also more heterogeneous. So even though this is an immortalized cell line from this patient, they still have pretty heterogeneous behaviors, which is good in some ways because that's what we might expect if you looked at clinical samples, but it also sometimes gives you really large error bars, unfortunately. And this was very exciting. This was a part of my early work as I was starting to grow my career. But the problem with using fibroblasts to generate strain in 3D models is that they're doing other things. So they are secreting matrix proteins. They're secreting matrix remodeling proteins. They're secreting cytokines and growth factors. So I created a pseudocell system where I took magnetic beads and I coated them with thrombin. And when we embed that into a fibrogel, and we do that with cells or with our blue tracer beads, and we put a magnet next to it, what happens is everything moves. And it moves as if a calf was there making those matrix deformations. So it is an equivalent strain without any confounding factors like VEGF, for instance. And we use that to actually, as a proof of concept, we put these magnetic beads into 3D microtissues that only had endothelial cells. Endothelial cells will not form a blood vessel network on their own. They need a little bit of help. And what we found is that the mechanical stimulation from these pseudocells was actually sufficient to drive vascular growth in our systems. That's the EC only sample, and then we had a bunch of other fibroblasts. Won't go too far into the mechanism, but basically these don't make blood vessels, these make blood vessels. The SH-ROC and SH-YAP are inhibited, so they will not make matrix deformations, but the mechanical stimulation from the magnetic system from the pseudocells rescued their vascular growth in those samples. And the last bit of model development, I promise, and we'll start getting into lots of science. But so we use this microfluidic model, and it is 3 tissue chambers in series. So the blue regions are areas that we can put in matrix and cells, and the pink areas are fluidic lines. And there are 20 micron wide communication ports between our tissue chambers, which means that we can control the interface between different cellular compartments really tightly. Just to give you an idea of scale, this is the full device made in PDMS, and from here to here is what's shown in that top figure. So this is very small, and I have more than one time gone cross-eyed trying to produce a bunch of these in lab. But the nice thing about it is that we can control a bunch of different biomechanical features semi-independently. So each of these tissue chambers can be loaded with a different matrix and a different cell population at the start of the experiment. The interstitial flow can be controlled both top to bottom and left to right because each of those fluidic lines is independent. And this lets us set up things like a pre-metastatic niche or a primary tumor site and have them be separate at the start of the experiment to see how they interact. Using COMSOL modeling, we were able to actually show that this system lets us fully control the diffusion of soluble factors between those microtissue regions. So in this flow, which I call top to bottom, the red arrows indicate where our high pressure of media is, and so everything is moving down individually through the chambers. And if you look at those interfaces, which are the red circles in that top graph, there are very, very, very tiny peaks. But that scale is microns per second, so we're talking like 0.0001 microns per second worth of interstitial flow. If we do high pressure in the center chamber and low pressure in the side chambers, we create this outward flow. And now we can see peaks inside those red circles that correspond to fluid moving from the center chamber into those side chambers. There's then no physical force moving anything from here into this chamber, which means effectively we've washed out anything that's in that side chamber that's a soluble or secreted factor. So that's that cartoon. And we did this with fluorescent dyes to actually show that yes, we do get those really clear lines between our chambers when we set up flow top to bottom. And if I take that same device and then put DPBS in it in the center chamber only, for that outward flow, we actually get a washout effect. So now we're not seeing any significant fluorescence from any of our materials retained in any of the chambers. So this lets us start to begin to separate the idea of soluble cues or biochemical cues from biomechanical cues. And the idea is that even though both of those are important inside an actual physiological system, if we can study them separately, we might be able to uncover ways of mechanical communication that can be targeted and as a part of new anti-tumor therapies. Going from COMSOL modeling into cell culture, we put normal lung fibroblasts and endothelial cells in that center chamber. And then we loaded either our CAF control or our mechanically inhibited CAFs. And then we had NBFs, which don't deform the matrix and don't make blood vessels. And we added a constitutively active row. So row is a GTPase associated with contractility in the cytoskeleton. We did that outward flow regime, so all of the media is coming into the center and washing out. That also means the side chambers are experiencing the same amount of interstitial shear stress, and they are receiving the same secreted factors from those lung fibroblasts and endothelial cells in the center chamber. So those are both controlled for, and the only thing that's different is the ability of the fibroblasts to tug on that matrix right at the interface where those dashed lines are. And what we saw is that our calves still made more blood vessels, so this is now angiogenesis into that side chamber. The CA row cells were able to generate more blood vessels, so I've got arrowheads to help point that out, because I never know how good the projectors are going to be when I go to places. This one's really nice, actually. And if we look at the matrix deformations now, we see that we can actually observe that change in ability of cells to deform things around them in those interfaces. So all of the measurements for these graphs were taken in those communication ports, where the CAFs or the MBFs met up with the endothelial cells. And we see higher deformations from our CAF EV, decrease from knockdown, and our CA row cells also have an increase in matrix deformations. So this is probably the last experiment I did by myself before I was fortunate enough to hire a PhD student named Bronte Johnson. So we could really start to investigate the mechanism that actually controlled this. Everything so far has been the model development phenomenon description showing that we can get mechanically driven angiogenesis. But now we want to know really what's going on and can we target that to shut it down. And so we want to look back at that VEGF and VEGFR2, which is the receptor type 2, which are key drivers of angiogenic blood vessel growth. VEGFR2 is also expressed on most epithelial tumor type cells. So breast cancer and ovarian cancer cells themselves have this vascular endothelial growth factor receptor on them. And like I said earlier, these drugs show limited improvements in patient outcomes over time. In fact, there have been some recent decisions by the FDA to pull some of the anti-VEGF drugs from certain types of cancers because they're just not providing enough help. VEGFR2 has two other family members, which is VEGFR1 and VEGFR3, which are related to either angiogenesis or lymph angiogenesis, but VEGFR2 is the primary driver. And what happens is that the ligand VEGF will bind to this receptor. The receptor tends to dimerize, which is why there are two of them together. And it is a receptor tyrosine kinase, so it has a lot of sites on the inner cellular region that can be phosphorylated in order to create a signaling cascade. And what we really looked at is some of the phosphorylation sites of VEGFR2 are tied to known mechanotransductive pathways. We know that SRC, PI3K, and AKT are sensitive to mechanical strains. We know that the MEK-ERK pathway is sensitive to mechanical forces. And that's related to certain phosphorylation events on this receptor. So certain activation types tied to certain downstream pathways. And we really wanted to explore that further, especially because we found this paper where they had looked at all of these different phosphorylation sites on VEGF. So all of the different possible activation places. They gave it soluble VEGF, which is that VS, or bound VEGF, which is the VB. So they basically attached that ligand to the matrix. And when that bound ligand attached to the receptor, you would get activation at 5 minutes. But I want to highlight these two over here. So here you have 5 minutes that are the same, 15 minutes it's gone away. 12:14 was really interesting though because the soluble and bound both activate at 5 minutes, but there's a difference at 15 minutes. So at 15 minutes, this receptor is still activated when presented with a matrix-bound ligand. And that suggests a mechanoactivation or mechanoreceptor type behavior. Bronte took that and ran with it, and she took human microvascular endothelial cells. This is an immortalized cell line. We have a flex cell system, so you put the cells in a monolayer, put that in an incubator on top of a vacuum post, and essentially when the vacuum is activated, it will stretch the membrane the cells are growing on. Do this in a very controlled fashion. So we used 10 hertz, which is close to the same thing that our calves were generating, and we used 10% strain elongation of 0.3 hertz, which is respiratory And then she either gave them no VEGF or she gave them a ton of VEGF. So 25 nanograms per ml is a large amount of exogenous VEGF to make sure that we were really hammering this receptor to get it activated. And she did Western blots, and what she saw is that we did see some increases with strain alone in 12-14 phosphorylation, that specific tyrosine, but it also worked with a combination of strain and VEGF, which her working hypothesis is that this actually is a really great model for the tumor microenvironment, because we know inside the tumor microenvironment the CAFs and the tumor cells will make VEGF, and we know there are these extra strains. And so she kept this kind of combination treatment in a lot of further studies, and then looked at inhibition of that. So SU5416 is a receptor tyrosine kinase inhibitor, blocks VEGFR2 signaling. And what we saw is that mechanical activation, so when strain is present and VEGF is present, there's some depression, some lowering of the overall phosphorylation. If you look at 1054, there's actually more phosphorylation that's occurring when these cells are stimulated with strain, even in the presence of this VEGFR2 inhibitor. And we thought that was really interesting because if this is not fully suppressing our activation pathways, why is this inhibitor used in studies? And is that why it's failing to make a difference clinically? She looked at this in other cell lines as well. So these are HUVECs. These are now primary umbilical vein endothelial cells. She did the same treatments. She still saw a lot of activation with strain and VEGF at both sites. And when we go in and we look at kind of total VEGFR now, and not just our phosphorylation, the total VEGFR increases in the presence of that inhibitor when strain is present, which seems very counterintuitive when you first look at it. Why would the receptor go up if there's an inhibitor? And we think that's a couple of things happening. The first is maybe the cell is trying to compensate because it does know that inhibitor is there. The other thing that is really interesting about VEGF signaling is that you don't want it to run rampant everywhere in your body all of the time. So it has a degradation protocol. And what happens is normally when VEGF is activated, the cell will internalize that receptor and degrade it so that it can't create a feed-forward signaling loop. But when you have strain and this inhibitor present, it looks like that degradation is actually blocked. And we've started to see this in some other cell lines as well that we've worked with. These was all 2D studies. This paper came out right at the end of 2023, so Florence started to kind of work on the next steps now. But she also wanted to look at things in 3D. And so this is that magnetic bead assay. I think I skipped a slide, hold on. There we go. Yeah. So this is that magnetic bead assay now. We've got cells embedded in it, a rotating magnetic field underneath. Again, she gave either VEGF or no VEGF treatment and vehicle or SU drug treatment with and without mechanical stimulation, all of the combinations of controls. She's done all of them. And up top, we have pictures of microtissues with blood vessels stained in red. And down here, we have length of blood vessel per area analyzed. And the biggest thing that's important on this graph is kind of these last two. When you give VEGF and vehicle, no magnet versus magnet doesn't make much of a difference with this cell line. When you give VEGF and the inhibitor, there's not actually a significant decrease in the amount of vessels grown when there is a magnetic force present. So in other words, even though we're inhibiting VEGFR2, in this 3D system, when the inhibitor is present, we are not substantially, significantly decreasing vascular growth. Now, this model doesn't have any flow in it. This is a static model. And blood vessels are meant to have flow with them. So Bronte did the same experiments with our microfluidic system. So now we have CAFs on one side, NBFs on the other, lung fibroblasts and endothelial cells in the middle. We use the lung fibroblasts in these studies because they just make really pretty normal blood vessels. So that we knew we could get a nice vascular network and observe angiogenesis from that center chamber outward. And for our vehicle samples, NBFs don't promote angiogenesis, CAFs do, that's the kind of wavy bars. And when you give it the SU5416, and this is outward flow, so it's diffusing everywhere, it's affecting every region in that micro tissue device, the CAFs still make a lot of blood vessels. The CAFs are still producing angiogenesis, even though there's SU5416 as inhibitor everywhere. She did the same experiment with magnetic beads, so that we could entirely get rid of talking about fibroblast, NBF and CAF secreted factors. And we see a significant increase in angiogenesis with the magnetic bead stimulation, with this pseudo-cell mechanical force, without any exogenous VEGF and without any endogenous CAF secreted factors. So Bronte's now, she's going to graduate in the spring, most likely. Everything looks like on track for that. But she has started to work on mutating the VEGFR2 receptors at these tyrosine residues to see if we can determine exactly how the mechanical forces are driving these changes in signaling and how that affects angiogenesis. About the same time all that was going on, I recruited a new student named Shalaria, and we decided we actually wanted to do some cancer research in my cancer research lab. You know, I thought it might be a nice change. So we focused in on triple negative breast cancer. Triple negative breast cancer is one of the most aggressive subtypes. Triple negative means it doesn't have the estrogen, HER2, or progesterone receptors on it, which are the three receptors that typically get targeted in a lot of breast cancer drugs. So in other words, there's not a great molecular target for this, which means it's really hard to find a drug that will help patients. And I told you that VEGFR2 is aberrantly expressed on these epithelial tumors. This is some data from the TCGA, where we looked at patient survival for triple negative breast cancer patients with respect to their VEGFR2 levels. And the higher the VEGFR2 expression, the decreased overall survival these patients have. So people are still looking at anti-VEGFR2 drugs for triple negative breast cancer. It's still under development. These are a couple that have been recently published in preclinical studies, but we're still seeing that only maybe 2 to 3 months of progression-free survival increase. So we took our model triple negative breast cancer line, which is MDA MB231. This will be abbreviated 231 in a lot of other slides. We did have VEGFR2 in them. We decided to go ahead and do shRNA. So our scramble has a lot, our knockdown has very, very little, and then MCF7 is ER positive breast cancer line. So that's kind of a built-in control we have now. So we have our triple negative line that has VEGFR2 expression endogenously, and this ER positive that does not. And as a starting point, we went back to our flex cell. We put our triple negative breast cancer cells on those plates, strained them for 5 or 15 minutes, and did the same vehicle or SU5416 and or VEGF and or strain. Again, all the combinations. These blots get very big at some point. There's a lot of data in these files. But the important thing, just looking at our Western blots, is here's our phosphorylation at tyrosine 1214. So that mechanically sensitive site inside VEGFR2. And with no strain, we don't have very high levels. And even with 5 minutes of strain, we see a dramatic increase in the 231 phosphorylation at this site. And when we go in and quantify that, what's even more interesting is that in the combination of VEGF treatment and strain treatment, there's no difference in activation between our vehicle group and our inhibitor group. So again, this is suggesting that something about either strain by itself or the strain specifically in combination with VEGF is turning on the receptor in some way that the drugs aren't blocking. So we think this might also explain why when TNBC recurs in a patient, the metastatic, the recurrent disease is even less sensitive to VEGFR2 inhibitors than the primary tumor. I also just wanted to show again, I talked about that degradation pathway that we think is messed up when you strain these cells. That same behavior is also seen in our breast cancer cells, where when you have strain, even though the inhibitor is present, you get large amounts of VEGFR2 expression. in these cells. So we're not blocking either the activation of the receptor, nor are we blocking it, and we are blocking its degradation, which means it could signal more. From a functional standpoint, we next moved into looking at proliferation of these breast cancer cells. So these are Z projections of 3D microtissues of the 231 cells with either no beads and VEGF or no VEGF and beads. And the red is Ki-67, so that's a marker of cell growth and division. And what we saw for the wild types is that when you have the magnetic stimulation without VEGF, we had a very high level of proliferation in these cells. However, when we moved into our, the title is incorrect, but when we moved into our scramble and our VEGFR2 knockdown cells, and remember we have about an 80% or so knockdown at the protein level. Red is again Ki-67, blue is just dapping. And what we actually see is kind of across the board, even before we started doing our analysis, it really looks like there's less proliferation happening in the SH VEGFR2 cells. When we ran through our quantification, that didn't seem quite as present. But the important thing to take away from that study is that the VEGF stimulation with mechanical stimulation made our scramble cells proliferate. Nothing really changed the SH VEGFR2 cells. So it didn't matter how much VEGF we threw at them, it didn't matter how much mechanical stimulation we threw at them, their proliferation was relatively flat across the board. Shalaria then did these really large migration studies and proliferation studies inside our microfluidic model. She started with the TMDC inside that center chamber and either CAFs or MBFs on the side chambers. We did tag the 231s with a TD tomato so that we could watch real-time migration. We actually tracked it over the course of 2 weeks. And we had that media flow in that outward position again. So all of the media was coming into where the TMDCs are and then flowing outward into the fibroblast chambers. At day 14, we fixed everything and we took a bunch of pictures. In our top image, the red is those TD tomato tagged 231s. In the bottom image are the side chambers, and everything has been stained for Ki-67, so that proliferation marker. So what we're looking at in this case is proliferation of all of the cells that happen to be in that side chamber. When we quantify that migration, our TD tomato 231s, there are more of them in the chamber with the CAFs than there are in the NBFs. This graph, it says normalized. It's normalized to the total red fluorescence inside the center chamber. So sometimes microfluidic devices work fantastically and you get great pictures, and sometimes you have maybe a little less cell growth. Cell cultures like that, if you haven't done any of it, sometimes you just roll with it. So the CAFs seem to be promoting more migration and or proliferation because there are more cells there. And when we look at our proliferation, so this is normalized again to that RFP, there is more proliferation in the chambers that contain CAFs than NBFs. And we'll caveat this data and say that this is both fibroblasts and tumor cell proliferation, so there's overall just more proliferation in the CAF chamber. We're working on all of the controls to make sure we can really separate out the signals to know which cells are dividing. When we do the same experiment, but now instead of using the TD tomato tagged cells, we use our scramble or our VEGFR2 knockdown cells. We see the same patterns. There's overall more migration into the CAF chamber than the NBF chamber. There's overall more proliferation in the CAF chamber than the NBF chamber. And the migration follows the same kind of trend with the VEGFR2 knockdown migrate a little less. This was a pilot study, so we're working on increasing these in numbers now. But it looks like there's more proliferation in our knockdown devices. Again, though, this is not corrected for the presence of those fibroblasts. So what we actually think is happening, based on our other proliferation study, is that if the knockdown cells are not proliferating as much, that means there's more resources for the CAFs and the NBFs in those chambers to be proliferating. this is the same data, but now we've normalized total cell count in the side chamber rather than total RFP. And so what we can actually see from this is that there seems to be a pretty stark difference in the number of scramble scramble TMBC in the side chamber compared to the number of knockdown, just from a percentage standpoint. So basically there's more CAFs present in the side chamber of the knockdown devices relative to the overall cell count. And so kind of our preliminary takeaway from this is that we are somewhat depressing that ability of the VEGFR2 knockdown cells to migrate. And while total proliferation has decreased in our NBFs for our scramble and our TD tomato cells, there's some fun mechanistic things we still need to work out what's going on in those VEGFR2 experiments. So hopefully you've followed through some of our abilities to investigate and interrogate these biomechanical forces within a 3D tumor microenvironment model. And again, in biology, things are complicated. It's probably a combination of biochemical and biomechanical forces that drive tumor progression inside human beings. But we can't study that easily, not in humans, and we can't study it easily in mice either. So we need these kinds of systems where we can separate out our flow, our soluble factor diffusion between regions, our cell phenotypes, so that we can really start to answer these different questions. How are they related? How are they linked? And then can we promote or inhibit specifically? I didn't show any of the data today, but we actually have a pro-vascular project in the lab where we want angiogenesis to happen because we're starting to talk about tissue engineering models as well. So in that case, all of the stuff that I presented today that we want to shut down, we'd want to flip that switch the other direction and use it to actually create a controlled vascular growth inside a given space. The other thing that we've shown is that while VEGFR2 can be activated by mechanical strains, by tensile forces, the inhibitors that are currently out there, at least the ones we have screened, and I showed this one today, don't block that effect. So the mechanical activation of these receptors may or may not be inhibited by our standard drugs that have already been developed. We also saw the same mechanical activation inside TMBC. I've got an ovarian cancer project in the lab, and if we ever have some extra antibodies, I'm probably going to have them screened for that mechanical activation in those cells as well. And while the knockdown of this will inhibit some of the migration, it doesn't seem to be, it's not like a tenfold factor, it's just a small effect. Shalaria literally sent me the data for the magnetic bead in the device studies with TMBC yesterday when I was trapped in Charlotte Airport. So I have not had a chance to look at that yet, but we are doing those studies to eliminate CAF and MBF factors and see if we get that mechanically driven migration with the bead system. So thank you again for having me. I am very fortunate in that I have built up a really great group of lab members despite starting my lab during COVID. I showed some of Bronte and Shalari's work today. Miranda is the one who does the ovarian cancer project. Vaishali is the one who does the pro-angiogenesis project. I also want to thank my collaborators and our funding sources. And just a brief disclosure, I do part-time consulting work for this company called Surflux. I didn't talk about any of their stuff today though, so don't ask me about them. They have a lot of weird NDA rules. But thank you again, and I'm very happy to take any questions.
Okay, so questions from the audience?
One question. In the study where you're moving the cells around with a magnet, how can you account for the convective transport of getting those free ligands, closer to the receptors just because you're moving them around?
Yeah, so this is one of the things we're actually playing around with now. Currently our magnetic stimulation is just an orbital rotator plate that I've wedged into the incubator. And so it's a cyclical magnetic force. We think that the ligand diffusion, any soluble factor diffusion in those systems should still be washed away ultimately, basically sink out into those extra fluid lines. we're trying to work on either an siRNA or a CRISPR system to knock out the VEGF gene itself so we can get rid of paracrine signaling. The other work that Vaishali has done is she's done a bunch of cytokine panels to really look at the difference in expression. And so the nicest thing I can say is that there doesn't seem to be significant differences in the CAFs and MBFs in the primary angiogenesis ligands. So even if there are some present, we're hoping that it's equivalent kind of across the board.
So the cells are already activated with the VEGF, and then free ligand is removed, and then you're looking at the mechanical stimulation after the chemical stimulation?
In these studies, it's probably a competitive feature with the ones that have the CAFs and the MBFs, if a ligand happens to be close enough to a receptor versus if that cell is passed over the magnet right at that moment, you know, when that receptor, I'm not sure which is the stronger bond, if that makes sense. Like, is the mechanical stimulation a stronger force, a stronger result than a ligand stimulation? We haven't quite teased that out yet.
Okay, over there.
So, how you plan to implement your research without into the clinical treatment in the case?
That's a good question. So the question was, how do we plan to translate this into the clinic? The very first thing we're actually doing is preclinical studies with mice with an orthotopic breast cancer model. We're simply going to try treating these mice with not just chemotherapy, but with also basically an anti-contractility agent. So things like Fasudil or verteporfin are already FDA-approved drugs that target contractility. And we think that this might be like an adjuvant effect where we can combine that with currently used clinical treatments to block this mechanical signaling. Yeah.
What research have you done outside of breast cancer?
Yeah, so the question was, what research have we done outside of breast cancer? So I talked a little bit about Miranda's project on ovarian cancer. She actually has done a really great study where she showed that, so as ovarian tumors progress, the hyaluronic acid content in the tumors changes. This also is related to patient age. So hyaluronic acid is a matrix protein. It's present a lot in your brain, actually. It's what makes your brain really squishy. But it also makes epithelial cancer worse. So it makes breast cancer worse, it makes ovarian cancer worse, it makes glioblastoma worse. And what Miranda was able to do is show that there's a dramatic difference if there's HA present in the microenvironment versus if HA is present as like a soluble or serum secreted factor. She showed changes to the CD44 signaling pathway that were related to primarily proliferation and then response to chemotherapy. And again, it's kind of this idea, what she showed was that when HA is in the matrix, it provides more of a mechanical cue, and that mechanical cue increases the cell's resistance to chemotherapies. So that just came out very recently, mechanobiology and medicine. Yeah.
I do have a question. So at the end of your presentation, you briefly comment that you're running some projects that would require angiogenesis. So I'm very curious. Can you disclose what those projects are?
Yeah, so that's another one that actually we just got the first part of the project published on cadherin signaling. So we have screened pro-angiogenic and non-angiogenic fibroblasts in our microtissue models for that ability to deform the matrix. And then we've characterized their cadherin expression levels and co-localization of those cadherins in co-culture and in single culture. So Vaishali's really shown that it looks like there's a strong correlation of OB cadherin as a marker of a fibroblast that might be pro-angiogenic in a way that we think there might be heterotypic cadherin bonding between the endothelial cells and the fibroblasts that's helping to create that mechanical strain to grow blood vessels in some cases.
Okay, any other questions?
Another question. So you chose triple negative breast cancer as a model, but really only one of the tri parts is associated with the VEGF, the vascular growth, right? So what was the rationale for that choice?
So twofold. The tri part is actually ER, the estrogen receptor, progesterone receptor, and then HER2. So VEGFR2 is not in that triple part. It looked through a lot of literature to really find that the most common places that the VEGFR2 aberrant expression was being reported was in the triple negative breast cancer cases, and less in some of the receptor positive cases. And then when I did like that, the TCGA survival curve data, if you include all breast cancer types and look at VEGFR2 expression, the curves level out together. There's basically no difference for the hormone positive receptor survival with respect to VEGFR2 expression. The other thing about it was that because there's not a molecular targeted drug for it, I felt like this was an area where we could maybe make a difference. It has the worst overall survival rate.
Right, and part of that is when it goes to the brain, it no longer, it's got a different
Right.
molecular profile. So,
Exactly.
and it's not even, it's almost impossible because you can't really biopsy it so well.
Right.
So there's really no way to know whether it has the same molecular profile as the primary.
Yeah, there's actually some really interesting work that came out a couple of years ago now looking at, they injected 231s and developed the organotropic brain, lung, and bone met models, and then they did genetic screening on it, and they actually showed that the cytoskeletal regulation is different for cells that wind up in the brain versus in the bone versus in the lungs. They didn't look at VEGFR2, they were looking at a lot of the stiffness markers. But so there does seem, there is something mechanically that makes them more aggressive in the brain versus how they are more aggressive in the bone.
Any more questions? Well, if not, that's the end of today's seminar. Let's thank the speaker again and Claudia wanted me to remind you there's a signing sheet, so make sure, for students taking seminar courses, make sure you're signing before you leave.